Number two, the criticality of catalytic machinery reduced the fitness of the active site mutation, meaning if you reduce the fitness, the virus can no longer survive itself. Then identification of nucleoside inhibitors became very challenging because of the mechanism of action, meaning because if you look at a nucleoside inhibitor, those are the substrate-like agents that would bind to the active site, be recognized as a substrate, get incorporated into the growing RNA chains that was being replicated, the viral RNA, and then cause a chain termination event. The problem there with nucleoside development is the nucleoside itself is not the drug, or the active agent, let's put it that way. It's the triphosphate of the nucleoside that turns out to be the active agent. So the nucleoside must be monophosphorylated, diphosphorylated, and triphosphorylated. And then that triphosphate must be a substrate for the viral polymerase to be incorporated into the growing RNA chain and cause chain termination. So it needed to be actually a substrate for 4 enzymes. And so that made the complexity of the medicinal chemistry very difficult, and the search for agents, very challenging. However, in 2004, we identified this molecule known as PSI-6130. It's a 2'-fluoro, 2'-methyl cytidine nucleoside. This molecule was shown to be quite potent, minimally potent against the virus, about 4.5 micromolar of EC90. What was interesting about it, though, was very safe. Now, nucleosides historically are very toxic agents and have been challenging to develop drugs for because some of the significant toxicity associated with are anemia, right, and bone marrow toxicity. But this drug seemed to be very, very clean. So we took, but the problem with this drug was metabolized, heavily metabolized. So the cytidine base was metabolized to the uridine base and ultimately produced a very high percentage of the uridine in the animal species, whether it be monkey, rat, or dog, or in fact human. That uridine turned out to be completely inactive. No activity whatsoever against the viral polymerase. So we ended up seeing if we could bypass this metabolism by making a prodrug, and ultimately made this molecule as a 3', 5' diisobutyrate ester prodrug. And in 2005, we saw improved bioavailability in animal models. And we saw somewhat reduction in the production of this uridine inactive metabolite. We took this into the clinic. The molecule showed efficacy, so we were gratified with that, but we still had a PK problem, meaning that we had a very high drug load, so we were giving about 1.5 grams of drug, BID, so twice a day, so that's 3 grams a day. And this drug also had still produced a very large amount of the uridine metabolite. So we then went back and re-engineered the program. And we actually took advantage of this inactive uridine metabolite. Because one thing we found out about the uridine metabolite was in fact that the uridine triphosphate itself was very potent against the viral polymerase and also had a very long half-life in the viral hepatocyte, cell. And that means that we could possibly generate a once-a-day dosing agent because of the existence of the long-life triphosphate. But the question was, why was the uridine nucleoside itself inactive? And it turned out because the uridine nucleoside was not a substrate for the first phosphorylation step. And what we ended up doing is applying this technology, phosphoramidate technology, for bypassing that non-productive phosphorylation step and allowing the next metabolic steps on di and triphosphate to ultimately make the triphosphate of the uridine, which was active. We also surmised at the time, actually, that we could use this phosphoramidate prodrug technology as a liver targeting strategy, taking advantage of first-pass metabolism. Meaning, the first organ that this drug would see once it got through the GI system would be the liver. And if you could take advantage of the metabolic processes within the liver, you could ultimately achieve a liver targeting strategy, because once the triphosphate is made in the liver cell, it will never get out because of the highly charged. And in fact, this did work. We took this into phase 1 clinical trial. We saw this proof of concept of 400 milligram once a day dose. We saw 2 log reduction in HCV RNA, and it's also turned out to be very safe. So we had this molecule in hand. The problem was that it was a mixture of diastereomers at the phosphorus atom. No one at the time had developed a method to asymmetrically make chiral phosphoramidase at the time, or actually been able to synthesize the unique diastereomers. We were able to ultimately do that, develop a technology that allowed us to do that, and ultimately made this molecule, which we call PSI 7977, which was sofosbuvir. So sofosbuvir had these really unique properties. It was completely pan-genotypic. It had this very high barrier resistance. In fact, there was only one resistant variant, the S282T resistant variant, but that resistant variant only had about a 10 to 15% fitness rate for that virus. This molecule could be combined with all the other mechanisms of action I talked before. It had unmatched safety profile. This was one of the things that really surprised us most about this molecule, that surprised me, was the safety profile. We could give grams of this drug to animals and saw no safety issues. And in fact, there's never been a safety issue observed in the clinic or in patients now more than 10 years on after the drug has been launched. It was also the first ever directed liver-targeting prodrug that was developed. So this particular clinical study, known as the Electron study, really changed the course of therapy for HCV. And what it was a small study, which we actually looked at a series of different cohorts, and one being PSI 7977, or sofosbuvir plus ribavirin. Now, ribavirin is another small nucleoside that was always given with its small molecule, always given with interferon. And when we combined that, we got 100% cures in the patient population. So this really changed the dynamics of the whole field of HCV and demonstrated for the first time that you can actually cure patients with a direct-acting antiviral that eliminated the use of interferon. This drug was taken into a phase 3 clinical development and ultimately demonstrated in a broad patient population that greater than 90% SVR cures across multiple genotypes. And in 2013, it became launched by Gilead Sciences as Sovaldi as the first interferon-free HCV cure. Now that wasn't the end of the story because it worked well in genotype 1, but wasn't the best. And so this drug was ultimately combined with an NS5A inhibitor known as ledipasvir to launch what is now known as Harvoni. And a number of other drug combinations follow with sofosbuvir as what is known as the backbone of therapy. And today, what you see, primarily the market is dominated by the drug Epclusa, which is sofosbuvir plus valpatisvir, which is a pan-genotypic agent. You give it one pill once a day for 8 to 12 weeks, and you achieve greater than 98% cure rates across all genotypes and all patient populations. So where are we today with regard to what has this done for HCV patients? It's the first interferon-free cure therapy. It's a single pill, fixed dose, short duration, 8 to 12 weeks, greater than 95% cure rates across all genotypes, even difficult to treat patient populations like patients with advanced cirrhosis, IV drug users, or all ethnicities. It's available now in pediatric forms. It actually has reduced the number of liver transplants significantly. In fact, today, transplant patients have an expanded access to livers because now they're actually using HCV-infected livers and kidneys in patients and then giving the drug to the patients after transplant to cure their HCV. So you're getting an expansion of the number of donors that are available for patients. It's also resulted in significant progress toward HCV elimination in a number of countries. So the countries of Georgia, the UK, Canada, even Australia, a number of countries have now moved very rapidly to use these drug combinations to ultimately achieve cures in their patient populations that have high incidence of HCV. But also very interesting is that direct-acting viral HCV cures are now associated with a 79% reduction in hepatocellular carcinoma risk in patients who have taken these drugs to date. So a big advancement for patients on HCV, and now the question arises, can you now eliminate HCV from the patient population, the global patient population? And that's possible with these drugs now, but I think what you also will need is a preventive vaccine which will prevent new infections, because you see new infections continue to rise, as I showed early on, continue to grow over time because of other things like illicit drug use that seems to be pervasive in countries like the United States. So, let's go on to HBV. So HBV is really a work in progress, okay? HBV is a very, very different virus. They both impact the liver. They both cause liver fibrosis, cirrhosis, and liver cancer. However, HCV is a DNA virus. It has a much more complex life cycle than HBV. The life cycle is shown here, and I'll go through it because it's relevant to the rest of the talk. So the virus enters the cell through the NTCP, or sodium taurocholate cotransporter polypeptide receptor. It loses its viral coat. Encapsulated what is known as relaxed circular DNA, enters the nucleus of the liver cell, and then uses the host DNA repair mechanisms, all the host DNA repair mechanisms, to form what is known as covalently closed circular DNA, or CCC. This then is then transcribed and translated to form adducts such as pgRNA, core protein, S antigen protein, E antigen protein. And so what happens is pgRNA, pregenomic RNA, is then encapsulated into using core protein. This is then the viral polymerase that's also made, is then reverse transcribes the genetic information to form relaxed circular DNA from pgRNA. This then could go on, obtain a viral coat, and then be extruded from the cell for further infection, or can actually recycle back into the nucleus, deposit more relaxed circular DNA to replenish the pool of cccDNA. The S antigen that's produced, so the S antigen is a protein that's produced in very, very high quantities in the patient. For example, there are, in a chronically infected patient, there are literally teaspoon-sized amounts of S antigen being produced in these patients. And what the S antigen is believed to be involved in is causing what is known as immune exhaustion. It overwhelms the immune system to the point where the immune cells no longer recognize the existence of the virus. And so this then is formed, and it forms subviral particles that then react with the immune system and cause this immune exhaustion phenotype. So the question is, how do you attack a virus like this? So back in 2014, 2015, we came up with a strategy that really is a combination strategy, as one would expect for any virus. And that combination strategy required us to do three things. It required us to completely suppress viral DNA replication and the replenishment of the pool of cccDNA in the liver. It required us to reduce dramatically the amount of viral antigen that's being produced, this antigen that controls the host immune response. And then third, we want to be able to reactivate or boost the host immune system to ultimately bring those T cells and B cells back into functioning mode to be able to now attack the virus in the infected liver cells. So there are three things we needed to do here. And so we set out to now develop agents in each of these three class areas. And so let's talk about the first area, so suppressing viral replication. Now, HBV, there are current drugs on the market that are being treated for HBV. These are drugs that are being used lifelong. We need to take them every day. And these are known as the nucleosides, tenofovir and entecavir, shown here. However, they inhibit this viral replication step shown here. So converting pgRNA to relaxed circular DNA. The problem is that this inhibition is leaky. So you still have production of new virus, you still have replenishment of cccDNA that's occurring. So the question is, what can you do to further augment this ability to inhibit the virus replication process? And that now looks at capsid-inverse capsid inhibitors. In capsid inhibitors, capsids are this core protein that exists around the pgRNA and relaxed circular DNA. And the idea is, can you inhibit the encapsulation of those pieces of genetic information? And so if you look at core protein, or a capsid, each core protein is about 183 to 185 amino acids. It's the building block of the nucleic capsid shell surrounding the pgRNA and the viral polymerase. So what's in here is the pgRNA and the viral polymerase that enable reverse transcription. Capsid in substance involves 240 copies of core protein in these dimeric units. And the proper assembly is really critical for replication to occur. So when you look at, in the late 1990s, early 2000s, there were several agents through screening that have been identified that actually inhibit encapsulation of pgRNA in HBV. And they were divided into two classes. One is class 1. Class 1 is represented by these heteroaryl dihydropyrimidines. And then class 2, which forms empty capsids devoid of pgRNA, are known as halogenated propionamides. Okay, so this class of molecules actually forms aberrant polymers. So you never form a capsid. The core proteins just form these aggregates of linear polymers. And here you actually form in the class 2, you actually form an intact capsid, but there's no viral polymerase or pgRNA inside. So we were very interested in this mechanism, and ultimately, in collaboration with a group at the Blumberg Institute, did a high-throughput screen and identified this molecule, this molecular class, known as the sulfonyl benzamides. Okay? They were modestly potent inhibitors of the viral replication. And so we took this molecule, AB423, into the clinic. And this molecule, although it wasn't ideal, we hoped that we could at least get proof of concept in the clinic. But it required BID dose and required a fairly large drug supply. The other problem is we had multiple metabolites of this. So once we got into the clinic, even in the phase 1A study, looking at safety and PK properties, we began to realize that this molecule really wasn't adequate to move forward. So we needed a second generation agent. And that second generation agent, we required improved potency to reduce drug load, improved PK, and improved metabolic stability. And that led to our identification of this molecule, AB506. Now, AB506 was substantially more potent than its predecessor, AB423. Now, how did we come up with this molecule from this molecule? And that turns out that there were two screening hits that we identified in the screening. The screening hits were the sulfamyl benzamides and then the ethylene dioxybenzamates shown here. And what we did, we actually did a sort of a homologation or a hybridization of these two, right? And ultimately came up with these bicycle systems, these chroman kind of systems, that once we did a little bit of SAR around here, these aminochromats showed some promising level of activity, so 2.3. So we were interested at this class at this point in time, based on this activity, and so we went around and did an exhaustive SAR analysis around this, looking at 4 key areas, right, of the molecule: the left-hand hydrophobic area, the bicycle modifications on each end of the chroman, and then the urea side chain, side chain. Ultimately, we made several thousand compounds in the space, and ultimately landed with AB506. Now, AB506 had great potency. It was active against all viral genotypes that we had access to in the laboratory, so essentially equally potent. It was also active against nucleoside-resistant variants. this molecule showed that it actually had the same mechanism of action as the propionamides, meaning it formed empty capsids. So you can see that in these gels where you see production of pgRNA, you see core protein being produced, you see capsid structure being produced, but you see no capsidated associated DNA and no encapsulated pgRNA. So these lanes are empty. So we're actually operating by this mechanism forming empty capsids here. What we also show, we got extra crystal structure, so you can get an extra crystal structure, co-crystal structure of these molecules. And AB506 shown in the aqua here, the sulfonylbenzamide shown in the red, and the HAP shown in the yellow over here, actually have somewhat different binding modes here. And this will be important later on to show. So we took this molecule forward, looked at PK in both mouse, rat, and dog, showed excellent PK across species, obviously very good bioavailability here, very good clearance. We took it in known as a hydrodynamic injection mouse model, where you take a HBV plasmid, a non-infectious plasmid, put it in the mouse, and produces viral protein. And what we could see is a nice dose-dependent reduction in HBV serum DNA over time. And in fact, what was very interesting to us was that we saw essentially equal activity in the serum, reduction of HBV DNA in the serum, as we saw in the liver. Now, and that's in contrast to nucleosides, would actually work much better in producing reduction in HBV DNA in the serum than is actually observed when we look at the liver directly. This molecule was taken into the clinic. And we saw us in the single ascending dose part of the study, the healthy volunteers, both a single ascending dose and multiple ascending dose, 5 days multiple ascending dose study. The molecule was very clean. We also then took it into HBV patients and demonstrated a nice dose-dependent 160 milligrams or 400 milligrams reduction in HBV DNA. So we're getting about 3 log reduction in HBV DNA at the 400 milligram dose, and also we can see the reduction in HBV RNA. However, one thing we noticed in the clinical study was that at day 18, in the clinical study, these are 28-day studies in HBV patients, what we noticed was that we were seeing an ALT elevation, a grade 2 and 3 ALT elevations in some patients, specifically Asian patients. We didn't know what this was about. Now, one thing you need to know about HBV or any liver viral disease is that you frequently get liver inflammation, ALT elevations in the liver. It's normal course for the disease itself. And so we weren't sure whether these ALT elevations were associated with the disease itself, or was it associated with the drug? Were we seeing some drug toxicity? So we went back and did a 28-day healthy volunteer study to encompass that longer duration. So if you recall, in our MAD, it was only 10 days. So we never got out to the time point, the 18 days where we saw the ALT elevations. And unfortunately, what we saw, this was in healthy volunteers, we saw an ALT spike in several patients, indicating that there was some liver toxicity associated with this drug. And therefore, this drug was discontinued from development. So our next question was, what is it going to be our third generation agent? We believed in this mechanism. Our strategy has always been, until we get a drug in phase 3, we continue to bring forward better agents, differentiating profile than the previous ones that can step in if something happens to a drug in the clinic. And so we set up with a product profile that looked like this. We wanted a differentiated chemical matter. And why we wanted a differentiated chemical matter was we weren't sure where the toxicity was associated with AB506. Some idiosyncratic toxicity associated with that in the liver that we could not explain. Because in fact, in AB506, we had chronic tox studies in rat and dog going out to 6 months and 9 months and saw no toxicities associated with the liver there at all. So we wanted a different chemical series. We want to improve potency. So we have improved potency will allow us to reduce the dose. That'll help in any potential toxicity associated with it. We also wanted to address some other issues that we identified with AB506. So in the AB506 clinical study, what we identified was there one patient that was a complete non-responder. And that patient turned out to have an T33N, excuse me, I105T mutations. So the mutation caused almost complete resistance to 506. So we wanted an agent that would address the resistant variants that we became aware of, or more becoming aware of within the clinical development programs. We wanted, and obviously we wanted a very efficacious molecule that would help us reduce dose as well, and then we wanted a favorable PK that gave us a once-a-day dosing regimen. So one of the things we did was actually look at the crystal structure work that we had done and that had been reported in the literature from other early work that was in the field. And what is shown here is, this is one of the class 1 inhibitors, so different phenotypes essentially than the others, although they all bind to the same active site. This is the sulfonylurea, and then this is AB506. And so what you see when you look at the crystal structure, you see that they all bind. One thing, there's no change in the backbone of the capsid, the core protein. You see they all have this binding region that puts a hydrophobic group in this hydrophobic binding cleft. They all have this hydrobonding network here, tryptophan and threonine binding here. That seem to be critical. And then there's other hydrobonding interactions that occur at the sort of eastern part of the molecule here, with this portion seeming to stick out differently from the other molecules. But to note, they all bind at the same site. So the question was, what was the pharmacore? The pharmacore model we ended up developing was this. It showed how this hydrophobic binding cleft. It had the hydrobonding interactions we needed at T128 and W105. It had this spacer that gave you to this other sort of unit that would require a steric piece that would bind in another pocket here, and some hydrogen atom donor receptor functionality. We came up with these molecules, these isoquinolines, that seem to fit this model. And first blush, these molecules had modest activity, but it was a starting point for the work that we thought we could do with this novel chemical series. And so we ended up doing an exhaustive SAR analysis, exploration with this. Again, 4 key regions: the hydrophobic region, the hydrogen binding region, this isoquinoline moiety optimization there. And we got extra crystal structures of these isoquinolines. And what we noticed was that they actually now bind somewhat differently than the other 3. So this is the HAP, sulfonylurea, the AB506, and now the isoquinoline. And what we found out was, in fact, it did achieve a different, unique binding mode. In fact, what you saw was that the hydrogen binding network was intact as before. The other molecules, you had the hydrophobic interaction and the hydrophobic cleft. But now we had this isoquinoline sandwiched between 2 aromatic amino acids, which was really quite different from what we saw in the other classes of molecules. However, this molecule behaved very much like a class 2 inhibitor, meaning it did not inform empty capsid structures, so no encapsulation of pgRNA, and still the capsid structure was intact. These molecules were quite potent, low single-digit nanomolar, active against hepatitis B and a number of cell lines. One thing this molecule did was actually, because of intrinsic potency enhancement, you were able now to engage that second mechanism. So as you may recall, in the replication process, there was the mechanism by which you form new virus that gets expelled from the cell, and then you got that recycling. That recycling mechanism seems that there's a sort of a tenfold difference in potency between those two mechanisms. So the lower you can get the first mechanism, the lower obviously you can engage the second mechanism more effectively and at a therapy, what we call a therapeutically relevant dose. So this molecule we took forward.